speaker. So henceforth with, so I conclude my talks. So on this session, so I congratulate all the students and I wish this program, so the 2 days program, a grand success, and I wish all the students should be benefited out of this program in their research as well as in their career. With these few words, I conclude my talk. Thank you one and all for this arrangements.
Thank you, sir, for your warm regards. I kindly request the chairman and the lead speaker of the session, Dr. Hamid Sulaiman, sir, Associate Professor, Department of Environmental Science, Sultan Qaboos University, to deliver the lead speaker address.
I'd like to thank the university to give this opportunity to come back. And as an alumni of the institute, it's a great moment to come back and stand here. So I just took a look at the university when I was here in 1989. I just want to talk about this before we go into the topic. So those are the days we have as a students like you. There's a lot of uncertainties. When I see you, all of you involved in organization of this conference, I think you are much privileged than us compared to what we did in 20 years here at the same campus. So we don't have these platforms at that time. You have a tremendous opportunity. And I also see the potential in terms of how you balance your teaching and learning research and your participation in extracurricular activities like this. That is something very good to see. And before I jump into the topic, I just want to bring into the notice our curriculums are built to bring in cognitive learning outcomes. So there is a shift happening in market to build two major learning outcomes when we spend time in schools and universities. The other two is socio-emotional learning and change in behavior and attitude. So majority of the curriculum focuses on improving our cognitive learning. So some of you may be not interested in participating in community activities, which is okay. But you learn a lot outside the textbook and curriculum through your communication and relationship with fellow human beings. Therefore, the two other major domains of learning are socio-emotional learning and how our learning outcomes change our learning behavior, at least when you are in the campus. So I just want to make this note because international organizations like UNESCO, they are focusing on these two later components because most universities, they did not address the issue of socio-emotional learning and behavioral change. With that, I hope that you have a tremendous opportunity to learn the two later parts along with your improving your cognitive skills. So we will talk about this later if we have a time, and I will jump into the topic for the sake of time. And this is a small study we did on the importance of vegetating our urban environment. So what is the context of this study? is this morning, the main speaker was talking about climate change as a very important issue globally. Climate governs everything. So, therefore, anything what we do today should be in concurrence with the change of climate because it will change everything. So, the small piece of work we did is very important, and it is the interface where science meets society. So our ultimate aim as a researcher is how we are going to change people's life. So definitely, directly and indirectly, it's going to affect people's life. So the context here, what you can see here, is a small study where social science research comes have an interface with the biophysical sciences. So as you can see from the title, it's the thermal comfort index in vegetative spaces of the urban environment. So because it's a scientific conference, I believe that we have to follow the structure of an academic presentation. Therefore, we talk about, I already started the introduction, and briefly about what is the objective of this research, then what methods we used, and small results we derive, and what is the implication of this research, at least to the local urban planning community and to larger policymakers. So because this topic may be very new to you, that you are sitting inside an air conditioned room, the thermal comfort index of all of us inside is very comfortable. You can ask and check people who are standing outside. They have a different perception of their comfort. So this is very important in terms of the micro level changes that is going to happen because of the climate change impacts. With the background in agriculture, you might have studied the impact of climate change from soil moisture loss. Evaporation rate started increasing, thereby the irrigation demands are going to increase in the years to come. And it touches upon every components of the ecosystem. But this study is specifically refers to the comfort level, which can be studied through the perception of the people, which can be shifted slowly without our notice because of the climate change. Why urban environment? Urban ecosystem is the most important human-made system which depends on energy, water, and other resources. Therefore, they are very vulnerable to climate change. So, to understand people's thermal comfort index in cities are very important. As people move from village to urban environment, probably because of the climate change, the rivers will take place after some period of time we do not know. So what we try to do, so I'm in the Middle East. So the climate is very hot. So 8 months, the temperature goes around up to 52 degrees centigrade maximum. And all the man-made buildings are air conditioned. So hardly you get outside. So every part that people move in are air conditioned. The life at the outdoor environment is very, very horrible. So because it's a hyper arid environment, we try to investigate the inhabitants' thermal comfort index sensation and preference to adapt to the increasing temperature. And we try to develop a regression physiological equivalent temperature model for the country which is specific to the city of Muscat. It's a very simple objective, and why this study was important, because this is the first study in that particular region. So with forestry background, my first degree and biophysical sciences, this is an opportunity to study the interface where your biophysical researchers touches the social science. Though we don't have a training on social sciences, the research trends are changing very quickly. As most of you know, if you're writing your thesis proposal at a PhD level and submitting that for a funding, the donor is going to ask you a question about what is the impact of your research on the community. If I'm not able to write something on that, no matter how complex your sophistication of research, funding becomes very difficult. So there we have to know exactly what is the implication of my research. So therefore, we try to develop this small regression model for that particular city where people are living. So it's a very simple study. It's a part of one of the bigger projects where in this study we measured the planted vegetation in urban green spaces and how much carbon they sequestered. In that note, we take into account how this vegetation is going to have an impact on these microclimate regime where the vegetations are planted in the city. So our study site on the right, you can see, this is how you see when you come to the Gulf countries. There is hardly any vegetation, and most of the vegetation is planted vegetation. And all the planted vegetation in the cities are for recreational and beautification purposes. And the policymakers have no idea about their ecological role. So therefore, it is people at the university to go and study them and tell them that it has other meaningful ecosystem services. So on your left side, these are how the vegetation looks like. You should not compare with what you see inside the TNAU campus, or at least in some of the cities around Coimbatore where the vegetation is. So these are mostly planted vegetation. So these are our two study sites. We want to compare what's happening between the study sites in terms of when there is a vegetation and how people perceive the comfort in terms of the thermal heat they are facing on the daytime, especially. The study takes into consideration the type of clothes worn by the inhabitants in the particular city. So if you want to study this in another context, so you are contextually, the clothing pattern your community is going to be completely going to change. So that makes the study very unique to that particular site or the community. As you can see, a lot of Indian based dresses here, because in Gulf there are 20 to 35% of the people are from three Asian countries, mostly from India. So we took into their consideration that what clothes people wear. And the study also involves measuring that microclimate meteorological parameters in the study site while we are doing the public survey, which basically builds into that model. And once we fit these data of the public survey and the meteorological condition in that area, you will be able to get what is the actual thermal comfort index temperature that majority of the people perceive as comfortable or uncomfortable. So once the data is collected, it fits into that particular software. So we put this, and with enormous pain, we are able to make this work, because in that part of the work, in that part of the world, social surveys are very difficult. So we took, this is the research design what we did. As you can see, the parameters recorded, and we are trying to get 200 respondents from each site, which took around more than 1 month, and it is a very kind of a one-to-one face-to-face interview with the respondents. Then we developed the equation, and we made some statistical analysis, that which I am going to show you in a minute. So this is the location, which is, as I said, you have a vegetated area and non-vegetated area. So this is that particular place where we conducted the respondent survey. And these are the temperature at the time of the survey. So now all these data goes into that model along with the social responses we got from the respondents from the city. So these are some of the demographic information of inhabitants in the survey. This is very important because it's a very small populated country, and 1/3 of the population are expatriates. So this is very important because it's a perception study. It's not a physiological study. Therefore, definitely what is perceived as comfort depends on how much time the people live in that area. So therefore, we took all these into consideration, and this is very important, that needs to be done at this particular level. So based on the response from the respondents, these are some of the data what we saw from the vegetated area and non-vegetated area. As you know, that social science has certain limitation, but overall, we found what we perceived, expected, has come out of the research. As you can see from here, people in non-vegetated area, majority of them, they feel that even during this, the study was done not in very extreme summer, they feel it is very hot for majority of them. And as you can see clearly that this is a perception response. So therefore, what majority of the research shows has certain implication for the whole population. So that makes the study very interesting. Therefore, though perceptions has certain major issues in biophysical sciences, but these are very necessary things that we need to study as a researcher. And these are some of the perceptions for our air temperature related to humidity, wind speed, and solar radiation in the vegetated area and the non-vegetated area. So clearly, the vegetated areas is where people spend most of the time, especially in that part of the world, for recreational purposes. Outdoor, as I said, people live 80% to 90% of the time indoor. What's the problem with that? They are hardly exposed to sunshine. There's a major issue of vitamin D deficiency in the community. So if somebody tells you, if you are living in GCC and they go and take a sun exposure, it's impossible because it's a scorching sun. So the only way people go out towards an outdoor environment is where there is a vegetation. And 90% of the vegetation is planted. All right. So this is the situation now, right? So now, in this particular study, which was attached to another study on urban heat island, we found the temperature in peri-urban areas are much lesser than the temperature which is in the Muscat city. So this is the whole problem with the change of climate, with particular reference to the urban environment, is the temperature is going up all the time. So people are moving for urbanization from villages to the city, and night temperatures are very high. I think I cannot explain to you unless in other ways you experience it. You don't feel that is it a night. It was very hot, right? So clearly then the implication of understanding this is now people are saying that they don't no more want to live in the city, they want to go outside the city just because of the urban heat islands in the city. So therefore, knowing people's perception in terms of thermal comfort index is very important, so that government should take a proper action, which are some of the implications of this study. As you can see from this, during the survey, we found that a majority of them, they feel that the vegetated area thermal comfort is acceptable. Though majority says that it's also comfortable, but it's very clearly no-go area for a non-vegetated area. So this is an hourly variation in the average physiological equivalent temperature. As you can see, there was a clear demarcation between a non-vegetated area and a vegetated area. This one particular graph, I think it is a evidence to show that the role the vegetated area plays in urban environment, especially in our study site. So there was a clear demarcation in terms of lesser temperature which are prevailing in a vegetated area. So converting vegetated areas which are naturally vegetated in cities like Coimbatore becomes more difficult in future if studies which are proving that some part of vegetation has to be maintained in the city. So we are looking for presenting this paper to the urban planners to seriously consider before removing certain patches of trees in a particular area before coming up with the development projects. So it is happening. I was just going through Coimbatore cities, some places what we visited when we were studying here. There is a lot of development. At the same time, equally, vegetation has been maintained. That's what my impression is, because I am visiting after 5 years. We used to move around. And one instance I want to share with you, when we are at undergraduate, we were with an organization called OSI. I think they are still on the field. In Araspuram, this cross, I forgot the name of the road. There's a one big tree. Yes, DP Road, yes. So they want to remove one big tree which is falling, which is basically smothering the billboard of a big outlet which is coming there. It's about to be cut down. Somebody phoned this organization, they come in and they are all on the street, and cutting was stopped. All right. So that was the thing which has happened even at that particular point of time. There is a lot of awareness. Okay, so for beautification purposes, there are certain things what we have to do, but we have to be also very careful about the age of the trees, which has been grown for a longer period of time, and their ecosystem services what they play in the city. So this is the academic scientific small contribution from this research. We developed this model and we found that the PET comfort range for Muscat city is 23.6 to 38 degrees centigrade. So basically, the maximum temperature goes around 50 degrees in the summer. But nobody feels this because everybody is in an air-conditioned room. So as I said, this is a part of the bigger project. But why I chose this to share this with you is just because the interfaceness of the study. So we have to train ourselves in some social science methodologies, which are very important as a researcher. And there is no harm in having a social science and economic objective in a hardcore science thesis. And that is very important because as a researcher, you have to know what is the contribution of your research to the community. So for academic interest, we want to check our studies result with other studies on thermal comfort index in different part of the city, including a city in India called Safidon. So now, what's interesting about this kind of study is wherever you study, you are going to develop that particular equation for that particular area. So that makes it more interesting. And therefore, after 10 years, if somebody wants to come and study, then you have to restart the study if our clothing patterns have changed. So basically, as we move on with change in culture in terms of how we dress and what changes are going on in the urban environment that keeps this kind of study very important at that particular point of time in the temporal scale. So these studies have been going on for a longer period of time. If any forestry or horticulture students probably venturing into this type of study or different type of study, you can also think of this as one of your objective and what is the impact on of your study on the public health in general. So what we found, we can humbly conclude in a smaller terms. As you can see from the conclusion, lower values of PET in vegetated area are indicative of impact of trees and its shading on thermal comfort of the residents. So the model established from the study have estimated the neutral temperature was 27.2 degrees centigrade, while the comfort range was between 23.6 to 38 degrees centigrade. So microclimate is a subclimate change at a micro level, at a macro level that happens at a city level, and city represents the country and the country represents the region and the region represents that part of the world to the global community of climate change. So what we believe from this small study is that these results will help the city planners, as I said, can adapt, to tell them to how to adapt by transforming outdoor urban spaces more comfortable for people experiencing heat stress. So with this small contribution from the part of our project, it takes a lot of attention to the people who are in community services to take note of this particular study. Probably they can influence the policymakers to consider such micro level changes happening in the city, in their urban planning and city planning in the future. With that, I will leave the floor if there is any questions to answer. Thank you so much.
And to find out the heat stress of an individual. And we are doing that different clothing, it has an impact on the heat stress. So cotton, it may be having different, in that way, whether you have conducted the research and how the heat stress is being monitored, only based on perception or any heat stress monitor is used for assessing the heat stress.
No, only on the perception.
Perception.
Yeah.
Okay.
Yeah. And cloth, as you said, we did
Yeah.
not look into what type of cloths, but generally what people wear, how the cloth look outside in terms of the name of the cloth, that is the basis. But as you said, probably yes. What is the cloth material and what's its impact of reducing the thermal comfort impact? That is a very interesting area to see, and people already started studying that. That's very it's the next step to do those kind of work. Yeah, number one is, as I said, they have only two outlets it's a hyper arid environment. People either go to the mall or go to the park. These are only two choices. Of course, people go to the beaches, but you have to suffer a lot of heat, scorching heat. So probably people go to the beaches only after in the evening when the sun sets. So, okay, now it's a very good question because if you ask the young generation in that, do you want to go to the mall or park? People definitely choose mall because malls are very cooler compared to, and you have a lot of things to buy and see. But the trends are changing with as people now wants to go in mall, you have very restricted places to sit. But in an open area and a green spaces in the city, you can take your food, sit there and spend time, so on and so forth. So the choice of this or that is something related to behavior. Now the government put these green spaces across the city as a recreational space. Now, for example, they have a proper zonation in the country. So it has a proper lung spaces for every unit of the district. So that spaces cannot be converted into any other developmental project areas. So now the number of people getting increased in park areas visit. So it is an implication for the policymakers to develop more parks in the city. So as you said, the change of behavior in terms of number. So there's a lot of people visiting a particular park and it's overcrowded. So that's a hint for the policymakers and urban planners to start another park, which is definitely very expensive for them because these are very highly, in terms of land values, they are very, very high. So that behavior driven changes will definitely influence the government in terms of greening more spaces in the city. But currently, I cannot answer your question because uh, we found more people in places where they find other values and rather than the recreational outdoor.
For a nice presentation on what is the project you are going to do, sir. So my question is that as we are in the urban area, many of the urban people are asking for the nutritional rich plants, that is the horticulture plants in their home garden, sir, as like of a jack or pomegranate like that, sir. Yeah, where there any research is conducted on what is the percentage of CO2 that each plant absorbs in the urban atmosphere? Like that, any research is going on there, sir?
Yeah, so there's a lot of studies going on in terms of how much carbon it's sequestered. Mostly these are studied in forest areas. Now these are translated into urban vegetation. So I was in Addis Ababa for a few years. So it's a highland, which is 2,100 meters above mean sea level. So a lot of vegetation there. And these studies were done in terms of how urban trees sequester carbon. At the same time, as to what you said, I mean, plants with nutritional values, what is their role in urban environment? So there are a lot of studies in terms of especially ecosystem services, which are related to carbon sequestration. But in terms of water retention, soil conservation, there are only few limited studies in urban environment. For example, Addis is like, for example, if you have a bike, people don't have a bicycle because when you get down, then you have to climb up. So there is a lot of runoff. So those studies are not done in urban environment because of due to so many reasons. But in forest ecosystem, the role of trees in terms of conservation of soil, moisture retention, the other ecosystem studies have been studied. The most but widely studied is the carbon and carbon sequestration and how much oxygen and potential it can release to the environment.
So my question is, what tip you will suggest is maybe a change from me or from ourselves will make your PET within the range or make our urban microclimate in the comfort zone about within your range. What, but one peculiar or specific thing, a tip that you will suggest is so that it will be changed in me.
Yeah, so that's a good question. What should I do is when there is no power, there is no air condition, don't complain. I'm just joking. So the see, that is the only thing, you know, our forefathers, they live in a very harsh environment. I'm not talking about only temperature. They live in a very kind of environment which is not comfortable, not only in temperature, in different financially not comfortable, I mean, health wise not comfortable. Those generation, they did not complain, right? So basically, that psychological adaptation is very important. So we have to be adapting to the change of mind and to be more tolerant about the change in the environment. So that are the adjustments we have to make as an individual. So basically, these are something you called as I said, socio-emotional learning skills and behavioral change skills. Okay, so clearly then that change as you said, individually we made, the community will change, probably we may not be complaining about thermal comfort index. So clearly then the government will be say that, okay, climate is changing, the biophysical environmental adaptation is happening. In agriculture university, we try to change the crop, hybridize them according to increase in carbon dioxide levels and heat. But human adaptation is also very important. So that adaptation comes from very simply in terms of changing our behavior. So if we start less complaining because we become mentally more strong, therefore there is no problem in the first place. Do you understand my point? So if let us say in 5 minutes all the power goes off, of course we can fan it like this, but how many of us are going to continuously keep on sitting and listening to next 2 hours, which is our main purpose as a student, right? So then clearly then that adaptability in terms of what is the actual goal. Okay, for example, the conference happens once in a year, then I put it in my schedule before 6 months, I am going to go to this session, right? 10 minutes switch, I mean, power goes off, it sweats, and the friends at the outside says that let's go out, then the adaptability is very weak. Yeah. So that's a very good question. So mentally, behaviorally, we change our thinking and adaptation. That will lessen a lot of problems in our life.
On behalf of the organizing committee, on behalf of the students, as well as on behalf of Tamil Nadu Agricultural University, we thank the guest speaker, Dr. Hamid Sulaiman, Associate Professor, Department of Environmental Sciences, for a nice presentation, as well as put forthing his views on the environmental aspects of this college, and also a very good kind interaction with the students, which has thrown a lot of light on the future programs of this environment. We thank you, sir, for the presentation.
Sir, for your presentation. It is now time for the commencement of oral presentation. This is a gentle reminder to the presenters. Every presenter will be given a total of 7 minutes, where you will have 5 minutes for presentation and 2 minutes for discussion. I repeat, you will have a total of 7 minutes, wherein you'll present for 5 minutes and you'll discuss for 2 minutes. We'll start with the first presenter, number 1.
Very good afternoon to one and all. And I welcome you all for my oral presentation on the topic an upshot of effect of pinching and paclobutrazole on pot presentability of zinnia. And this is a brief introduction about my crop, Zinnia elegans, which is a popular warm weather annual, and it belongs to the family Asteraceae and is originated from Mexico. And its uses ranges from use as a cut flower, or it can be popularly used as a bedding plants in landscaping, or it can be used as a potted plant. Its wide application is in butterfly gardens because it is an actual kaleidoscope of colors with different colors ranging from red, yellow, blue, red, yellow, orange, pink, and etc. Because of its color and its form, it attracts the butterflies. And to our interest, it is the first flowering plant that is grown and flowered in space research also. And this is my objective and treatments of the experiment. My objective is to assess the effect of pinching and paclobutrazole on pond presentability of zinnia. The main motive to articulate this objective is that zinnia is a very good bedding annual, but its application as a potted plant was comparatively low, maybe due to its disproportionate stature because of long internodal length and long plant stature. But it has a good potential to be grown as a potted plant because it is having more flower longevity and flower retentivity in the plant, and it is hardy to grow and availability in different colors. So for this purpose, I have selected my factors like pinching and paclobutrazole that makes the plant to be short and have a good form when it is planted in a pot. So here I have two factors, one is pinching and another one is paclobutrazole. In pinching, I have 3 pinching, single, double and no pinch, and in paclobutrazole, I have 4 levels, 50, 100, 150 ppm and no spray. So 3 levels of pinching and 4 levels of paclobutrazole, totally made 12 treatment combinations for my experiment. And this is the materials and methods. The place I've done this research is College of Horticulture, Hyderabad, and the design of the research is FCRD, factorial completely randomized design, and pot size is 10 in size and season is Rabi 2020. First of all, I have taken the seeds and planted them in a pro tray with 1:1 ratio of coco peat and vermicompost. And after 21 days, when the plantlets are ready to be transplanted, I have transplanted to them in a 10 in size pot with a potting mixture of FIM, vermicompost, coco peat and red earth. And this is a pot view after transplanting. So this is a pot presentability. It is nothing but how well or how good a plant can be visualized when it is placed in a pot of particular dimension. It was given by Conover in 1986. So he given this pot presentability score. So it will have some parameters like flowering, shape and form parameters. So this chart contains parameter, maximum points, description and score here. And first of all, let me say about flowering parameters. So in flowering parameters, we have number of flower per pot, flower size and color here. And the maximum point represents the maximum amount of the score which can be allotted to the particular parameter, and description is nothing but the characteristics which decides the score to a particular pot. For example, if a particular pot is having greater than 20 flowers, it will be scored 20, and if it is having 20 to 15, it will be scored 18, and so on. It is same for flower size and color also. And this is a second parameter, which is shape parameter. In this, we consider stems and foliages. And here we will see how self-supporting or how healthy, or if it is having any infection or diseases in the leaves. And accordingly, the score will be allotted to the pot. And third one is the form characteristics. In this, we will see plant height, plant spread and appearance as a whole. Here we will see if the plant is too height or if it is too low, or if it is feeling like overflown from the pot, and the score is given allotted, and the score is given accordingly. So total, the score will be given to a total of 100. And these are the results of my experiment. Here in the pinching, I have removed the terminal part of the shoot. And in paclobutrazol, we know it is an growth retardant, and it is a growth retardant, and thereby it will decrease the size of the form and makes it pot presentable. So by this I can say that P2R2, which is 100 ppm of paclobutrazol with double pinching, recorded the highest pot presentability score, whereas the least was accorded with the control treatment here. And these are the pictures representing pinching and paclobutrazol effect on the pot presentability here. Here we can see increased plant spread, and here we can see the decreased plant height here. And this is my conclusion. So hereby I conclude my presentation by saying that double pinching got highest pot presentability score among the pinching treatments with 73.28 score, and among paclobutrazol treatments, the 100 ppm treatment got high pot presentability score with 79.13 score, and among the treatment combinations, I have got like double pinching with 100 ppm of paclobutrazol got highest pot presentability score, which is 87.41, and least is control. Thank you.
That one. Good afternoon to one and all present here. I'm going to present under the theme 5. My topic is bio-earning of micropropagated Naipo and banana using bacterial endophytic consortium to promote plant growth and management of fusarium wilt. So introduction, you all know banana is the one of the second most important fruit crop in next to mango. It is have year-round availability, affordability, and it have different varietal range and nutritional value, and it have high medicinal value, and it have good export potentials. So these are the advantage of banana, but there is only one drawback, that is Fusarium axisporum species cubensis. So the first picture indicates the leaf hanging around the pseudostom, and second picture indicates the pseudostom splitting, and the third picture indicates the vascular discolorations. I collected this vascular discoloration. This is the Fusarium culture. They are subjected to microscope. This is the Conidia spore. So materials and methods, the crop is banana cultivar Naipo AB. Number of treatment is 7, replication is 3, design is CRD. So I have 7 treatments. So I collected 7 different kinds of bacteria from the Department of Plant Pathology. In that 2 bacteria only effective. So this is Brachybacterium paranglometrum. So this white color indicates the Fusarium axisporum, and this indicates the bacteria. So it inhibits the Fusarium axisporum. So this bacteria have somewhat effectiveness to the Fusarium. So the second picture indicates the Bacillus valigenis. So it have 40% of effectiveness. This is the control plate. You see difference. So this Brachypatria parangulatum, only half of the plate only Fusarium will grow. You see control plate, it fully grown. So these plates are subjected to semi-imaging. So these are the Fusarium conidia. These bacteria are colonized there. So this mechanism is called hyperparasitic behavior of endophytes on the mycel and conidia of FOC. So then I want to prove that whether this bacteria is effective or not. So I'm go for GCMS study. By using methanol extract, I go for GCM study. So in that well, I add 2 to 3 microliter of bacteria. This is the Fusarium culture. So that shows inhibition level. So I got two component. One is clindamycin and 5-hydroxyl 2-methylfluoryl. These two components is responsible for antifungal activities. So next I'm going for this, I take this two culture for further studies, that is biohardening. So these are how the procedure is biohardening done. So this conical flask I have brought, this have 30% of bacteria that are kept on the 48 hours shakers. Then they are mixed with hardened materials. So I bought tissue culture plants from outside, and I weekly once I will inoculate. So you all see this is the best treatment, that is Brachypatria parangulatum plus Bacillus fulgens. You see tremendous increase. Its plant height is about 21 centimeter. It is control plots. And also in best treatment, you see the root colonization was also observed. It have more root colonization when compared to control. So these are the semi-image of root. So this is the Brachypatria parangulatum. This is round space are the bacteria, and this is roots portion. So for that proof I done for semi-imaging. So these Bacillus valens are mostly rod shaped. So here the presence is that. This is the inoculated plant. This FOC inoculated control is there. So next secondary studying, I inoculate the FOC into the plants in treatment 2 and control. You see the difference. This control shows more Fusarium, that is severe distance. So I cut the pseudostome portion. There is only 95% of inhibition. Here fully inoculated control, it shows Fusarium symptoms. So what I conclude is, so identification of effective biomolecules. So this bacteria have growth promotion effect in banana plantlets, and root colonization also high. So it is one of the effective management of Fusarium by biohardening approach. As a bacterial endophytes.
Endophytes against the Fusarium.
Fusarium multis.
So what is the race?
Sir, this
Which
is
race? Which race you have worked?
race 1 only, sir. During its MSc, race 1 only confirmed, sir. Now 2020 only they identify race 4 in Indian conditions, sir.
Okay.
In Uttar Pradesh, Karnataka and Tamil Nadu, in 2000 only they like this work was done by 2020, sir. Race 1 only, sir.
So you have worked on race 1.
Yes, sir.
Now
Yes, sir.
the race 4 is very prevalent.
So now I am working in my PhD working also. This continuous only, sir. I'm working in race 4, sir.
Okay.
So we go for, this is only research studies, then only we go for field trial. So now we are checking only one field condition. So we have to select some hotspot regions of Fusarium. So in Tamil Nadu, in Theni area, granite cultivation is more. So nowadays race 4 confirmation is so. In Tiruchi they are NAIP 1. So like that we have to select the hotspot region. In that also we go for trial, and then only we will release. So nowadays in NRCB they are releasing on adaptation technology. It's called say no to wilt. So they are based on this microbial consensus. So in world level, it's one of the major who are the in banana industry. Threading is major. So they are focusing on how to manage it. So fungal chemical study, which is not effectiveness. So on the way, this bacteria, and I'm now doing in PhD actinobacteria. So this actinobacteria produce more antioxidant compared to bacteria. So I'm hope for if I get effective means it's very helpful for farmer field itself.
Good afternoon to one and all present here. Today my topic is on effect of organic manures and biostimulants on the yield of snake gourd. We all know that the vegetables are rich sources of vitamins and minerals for human diet. With increasing population, there is an increasing demand of vegetables, urging a great necessity for increasing the production of vegetables. The snake gourd is an important annual cucurbit, belongs to the family Cucurbitaceae, and it is originated in India. It is a monoecious annual herb, climbing by 2 to 3 branched tendrils. Regarding the nutritional value, the snake gourd is a very nutritious vegetable. It is rich in calcium, magnesium, phosphorus, magnesium, manganese and all. And it contains 86.2 kilocalories per 100 gram of edible portion. And it is rich in vitamin C, that is 30.5%. These are the health benefits of snake gourd. It helps in digestion, it boosts our immune system, and it helps in managing the diabetes, and it helps to maintain a healthy heart, and it stimulates the growth of new hair. Let's see why we have to go for organic. The intensive use of only chemical fertilizers for creating higher production led to the deterioration of the soil microorganisms and the aeration and all. So we have to go for this organic. The organic farming practices reduces the exposure to chemicals and pesticides, and it also improves the soil fertility, and it boosts our immune system. And the organically produced vegetables taste is better than the non-organic foods. And the farmers can reduce their production cost because they need not to produce expense to buy the chemicals and fertilizers. And it also ensures a safe and healthy world for the future generation. And then the methodology. The snake gourd variety used in the study was code 2, and its duration is 4 months. And it is a pure line selection from the local type of Coimbatore district, and it is a short type variety released by our TNAU. And the experimental layout was designed in the RBD, and it is replicated thrice with a 9 number of treatments, and the spacing provided was 2 meter into 1.5 meter. And here I listed my treatment details. The treatment one is control, that is recommended dose of fertilizer plus FOM 12.5 tons per hectare. And treatment 2 is audio plus FOM plus F2 microorganism. The ratio of 1 is to 500. And the treatment 3 is audio plus FOM plus seaweed extract in the ratio of 3 milliliter per liter. And the treatment 4 is recommended dose of fertilizer plus vermicompost alone. And treatment 5 is RDF plus vermicompost plus effective microorganism. And treatment 6 is RDF plus vermicompost plus seaweed extract. And the treatment 7 is RDF plus phosphobacteria. And the treatment 8 is RDF plus phosphobacteria plus effective microorganism. And the treatment 9 is RDF plus phosphobacteria plus seaweed extract. Coming to the results and discussion, the organic manures and biostimulants have been found to produce a positive effect on the yield parameters of snake gourd. Here we can see that the number of fruits per vine was increased in the treatment 6, that is by the application of vermicompost and seaweed extract, that is 3 ml per liter. This may be due to the combined effect of that vermicompost and seaweed extract that helps in the increasing the microbial population and also increases the respiration, so that the yield characters was increased. And the fruit length and the fruit girth was also found to be highest in this treatment 6 only. This is due to that effect of cytokinin, and the highest amount of potassium in the seaweed extract may be the reason for this increase in the fruit length and the fruit girth. And the single fruit weight was also found to be highest in this treatment 6. And then the fruit yield per vine and fruit yield per hectare was found to be highest in this application of recommended dose of fertilizer for vermicompost 5 tons per hectare and seaweed extract 3 ml per liter. This is due to the presence of that highest amount availability of water soluble nutrients in the vermicompost, and in addition to that the vermicompost releases the nutrients slowly, and it produces a nutrient throughout the growth period. And the net return and the benefit cost ratio was also found to be highest in this treatment 6. This is the field of my experimental field, and this is the comparison of fruits from different treatments. And coming to the conclusion, if vermicompost and seaweed extract were used for organic farming, our dependence on chemical fertilizers will be reduced, and it can be concluded that the application of audio plus vermicompost and the foliar spray of seaweed extract was most effective in enhancing the yield parameters, and it also increased the net return and the benefit cost ratio. So we can recommend this for farmers for increasing the production of snake gourd. Thank you.
Good evening, one and all present here. I'm very grateful. Thank to organizing committee for a selection of my topic for oral presentation. The topic is estimation of genetic variability and character association analysis of underutilized ornamental canali. This is my PhD, one of the part of primary pioneer study. Let's intro about the crop canna. The molecular sequencing analysis of plant taxonomy last 25 years, the APJ4, the angiosperm phylogeny group classified since 1998 to 2016. The canna classified separated from the order Gingiviales, the monocotyledonous. It is a single genus of canna family Cannaceae. It is one of the underground rhizomatous herbaceous perennial plant. The The plant having both flowers and the rhizomes used as the food additives, it is not fully underutilized for any food purposes in India. Recently, the canna has been recognized as 20 species studied based on the morphological and molecular analysis. And the picture shows the map showing the between the general distribution of the canna of 5 elemental canna species, for example, canna glauca, indica, iridifolia, and flacida. The state parsimony of state tracing biogeographic of canna species, the canna indica distributed all over the world of polymorphic characters. The nutritional composition of leaf, seeds, and rhizomes. Leaf generally and the nutritional composition present in ash content, fibers, carbohydrates, proteins. In rhizomes, same all carbohydrates, lipids, proteins. The high medicinal activity based on the quantitative analysis of biomolecules of secondary metabolites of phenols, uh, saponins, all are flavonoids like anthocyanins. The natural variability and uh, of morphological and anatomical process, and the presence of high amount of starch present in the canna starch instead of maize and cassava, it's used as the industrial food, baby foods. This is the leaf and canna seeds and rhizomes. The main objective of my hypothesis of this topic, the flowers is with the different colors used as the natural food colorants in the food and fabrics has the advantage over the synthetic dyes in human health. The second hypothesis of natural dyes are used for the treatment of various human ailments, for example, cancer treatments. The rhizome has the potential source for starch content. From this I have choose the objective to assess the genetic variability character association among the various yield attributing types of canna indica. The materials and methods I have designed for RBD with to collection of 12 different canna species with the different southern states of Andhra, Kerala, and Tamil Nadu, and Union Territory of Puducherry. This is I have cultivated the crop in the field of botanical garden. Next, the table shows the analysis of variance, the mean sum of squares of all the 23 traits of canna. India with different 12 accessions have a highly significant with 5% level. The performance levels of canna accessions based on the genetic parameters of 23 traits. Among all the traits, the flower yield per plant and rhizome yield per plant have the high heritability and genetic advance means as percent of it is highly influenced by the additive gene actions, not influenced by the environment. Coming to the genotypic correlation and the interrelationship between the 23 quantitative traits of canna indica, the accessions, the 12 accessions for compared with the rhizome yield per plant, the inflorescence are directly correlated with the rhizome yield and flower yield per plant, and also followed by the stem diameter and plant height, single flower weight also. The coming to the path coefficient analysis of quantitative trait size of 10 canna indica 12 accessions. The number of leaves per clamps related with flower yield and rhizome yield per plant. It is direct and indirect effect of the both yield. From the I conclude from this topic, the result revealed that among the 12 accessions 8 showed the higher flower yield per plant and rhizome yield per plant. It shows 329.9 gram and 891.8 grams. All the characters, plant height, stem diameter, leaf area, fresh weight of the rhizomes, number of tillers per plant, all the characters related, and it will be go for the genetic improvement of both flowers and the rhizomes yield per plant of the canary. Thank you.
Good afternoon all. I welcome you all for my presentation on evaluation of 4-year-old islanders excelsa-based agroforestry system in arid western Rajasthan. This study was conducted at ICR Kajri, Bikaner, Rajasthan. Arable crop production in arid region is highly unsustainable due to very low rainfall, very high temperature, poor soil fertility and wind erosion. So integration of trees in farmlands is the best option to improve the livelihood status of farmers in those areas. there are a few trees available in those areas, but they are very slow growing and economically not viable. So we introduced the Islandus excelsa based agroforestry system in those arid regions because it is a fast growing species. The wood is purchased for production of plywood in Sikar and Churu districts. It is high rainfall areas and the leaves are rich in nutrients and sold at 2 to 3 rupees per kg for fodder purpose. And in those districts, they are grown as a shelter belt or boundary plantation. So with this backdrop, we planted Islandus excelsa along with cluster bean at 3 different tree spacings. This cluster bean is grown for its seed, not for vegetables, and the seeds are used for the production of guar gum. And the trees are irrigated with a drip system and the crop is rain fed. At 4 years after planting, the trees were very taller at 6 into 6 meter spacing, which is on par with 8 into 8 meter spacing. And average girth was maximum at 8 into 8 meter spacing, and it was on par with 6 into 6 meter spacing. Whereas the trees shown very poor performance at lower spacing, that is 4 into 4 meter. This is the plantation area before establishment, and this is the overview of the plantation. And this is Islandus excelsa at 3 different spacings. And this is Islandus excelsa with cluster bean at a different growth period of trees. And this is Islandus excelsa with cluster bean at a 4th year. And we calculated cluster bean grain yield per hectare for all 3 spacings, and it was maximum at a 6 into 6 meter spacing, and which is on par with sole, which means there is no significant yield reduction in cluster bean yield at 6 meter spacing compared to sole crop. Fresh fodder leaf and branchwood production was maximum at 8 into 8 meter spacing, which is on par with 6 into 6 meter spacing. This branchwood is used as firewood by local people in those areas. We also calculated carbon stock and carbon sequestration, which was maximum at 6 into 6 meter spacing and followed by 4 into 4 meter spacing because of more number of trees in closer spacing. So at the end of 4 years, it was found that wider spacings showed significantly higher tree growth compared to closer spacing, that is 4 into 4 meter. And intercrop production was higher at 6 into 6 meter spacing, and intercrop yield is not compromised at this spacing. So it is concluded that Islandus excelsa can be grown at optimum spacing of 6 into 6 meter along with cluster bean without any yield reduction. And this Islandus excelsa based agroforestry system can sustain agriculture production, livestock production, and also increase the income of farmers through sale of wood, firewood, and fresh fodder leaves, along with carbon sequestration potential. By seeing this plantation, one farmer he planted Islandus excelsa in 3 hectares along with vegetable crops in his farm. On videos, sir, if time permits. Sir, HSC 368. No, sir. For that area, that variety is better.
You have mentioned about shelter beds.
Yes.
How many rows? So they are.
Sir, single row only. Mostly that high rainfall areas they are growing. This same arid region now only we are trying. Yes, sir. Complete field. That photo.
So it better. So row by row, and in between rows, you have the space in which you are raising this.
Sir, we evaluated 3 type of spacings.
6 by 6 meter.
Better. In between we grown cluster bean.
Have you measured the light intensity?
Yes, sir. That microclimate data we measured. Soil data also we measured.
Yeah, I really enjoyed your presentation, especially the videos of the site. I understand that you focused more on the crops and the trees themselves, but I'm curious, since you mentioned that it's a very harsh climate, did you observe the plantation to be benefiting the local biodiversity in some way, like maybe some species you saw there on site?
No, it is a full sandy soil.
Okay.
We didn't see any other vegetation under the trees.
Okay. No, I mean, in
No.
terms of,
During rainy season, weeds are there,
yeah.
and one grass also there. Brood grass, they used to tell. Otherwise, animal, they used to
You
nothing.
didn't see much. Okay. Thank you.
We will disperse for a short refreshment break. The next presentation will start by 4:35.
Good afternoon all. I am S. Aravindan from Center for Agriculture and Technology. I am going to present on my topic on implementation of modern molecular and electrochemical tools for the male and female segregation Palmyra pump. Palmyra pump are slow growing dioecious perennial tree. It's one of the most economical tree in India, which spread around 85.9 billion, out of which 60% of palm are distributed over Tamil Nadu, for which it is in 1978 it is stated as a state tree of Tamil Nadu. Palmyra palm is also noted as a tree of life, which gives which is a most economical multi-purpose tree. The importance of sex discrimination in Palmyra. As of now, in India, most of the palms are widely distributed. There is no proper adaptive cultivation in Palmyra due to the lack of sex ratio in Palmyra, which cause inadequate static in Palmyra production and its utility. What is the problems associated with Palmyra? Palmyra species are mostly identical in seedling stage until it produces first inflorescence. But the first inflorescence takes 12 to 15 years. So it's a very long juvenile period for Palmyra. So these factors has a main problem in farmers for implementing the Palmyra as a production due to its unknown sex in cultivation. So being able, so we are able to find a segregating of Palmyra. We started with the marker studies. On marker studies, we extracted 4 markers from literatures, which is 2 male specific marker and 2 female specific markers, and we analyzed the Palmyras of various stages. These are some markers we analyzed, but the markers doesn't show a prominent reliable results on sex segregation. So this shows the complexity in Palmyra sex discrimination due to small sex determining regions. So what we decided is we use another tool for sex discrimination Palmyra, and we use a cyclic voltammetry. Cyclic voltammetry is nothing but is a electrochemical technique for studying redox reaction. Redox reaction means reduction and oxidation reaction of a sample or a substance which is placed in electrode. And this is a cyclic voltammetry result. For a say, the cyclic voltammetry result is a duck-shaped plot. So we cut a Palmyra leaf into 2 by 2, and we placed as an electrolyte between the electrodes of a electrochemical workstation to see the CV, and we able to generate a pattern of Palmyra samples for male, female, and young samples. Here we can see we able to generate the pattern of Palmyra, which is more reliable between male sample and young sample. This is cyclic voltammetry data, but it is a irreversible reaction. In this graph you can't see the duck plot shape because here the oxidation reduction doesn't happen. It's so negligible. But our Palmyra is reacted to our applied voltage. So the materials responded to the voltage. The voltage is not directly related to the redox reaction, but could arise as a resistive effect of the electron-electrolyte interface. So on a note, we placed the Palmyra cross-section relief between the electrodes. As considered as electrolyte, we checked the specific capacitance of Palmyra for male, female, and young, and we get results of male and female shows moderately same specific capacitance, and we see the specific current density of male, female, and young. Specific current density, according to Faraday's law, is the amount of the substance transformed on applied current or applied voltage. So here the amount of substance may be ions. So here in specific current density, male and here also male and young shows moderately same results. So here I conclude by saying that in future aspects, the integration of marker study and electrochemical study has more give precious analysis of sex discrimination in Palmyra palm. Thank you. We first stand as a male and matured male palm and a matured female palm, ma'am. And we categorize the young plant based on the pattern generation and its specific capacitance and the current generated. We're going to categorize in the if this comes to comments, we're going to categorize into male and female.
Young seedlings, no?
Yes, sir.
So even after checking molecular studies,
Yes, sir.
you have to wait for until the flowering, no?
Yes, sir.
Then only we can confirm.
Yes, sir.
For study.
Sir, if in future if someone discovered a male specific marker, that time also we should wait for some more years for a first bloom. But by integration of electrochemical and the marker, we can confirm, sir. We can confirm by 80 to 90%. We can confirm it's male or female. Yes, ma'am. Specific marker, but the results are not reliable, ma'am. Both are amplifying at same base pair only, ma'am.
Yes.
RADP mark, only one RADP marker, ma'am. In 2007, one RADP is there.
Have you done? So the same type of problem is in papaya.
Yes, sir.
So is there any work done on papaya or date palm? Do you have any literature?
No, sir. In electrochemical workstation, this is the first time we are using in Palmaras.
So finally, you are now at present you will not be able to identify whether it is a male or a female in the
Sir.
end stage.
Sir, we can identify, sir. We can categorize, but should wait for some more years.
That's what I am asking.
Sir.
So
Okay, before we close, I think it's a conference for students. Now, with my experience, I just want to say something today in 2-3 sentences. You know, all your work in thesis ends up with a thesis examination. Is that correct? Okay, when the thesis examiner comes, he's going to ask you a lot of questions. Is that correct? Okay. Now, how do you prepare yourself to receive the questions? Is only when you ask the question, because it's a reverse psychology. Why we are not willing to ask the question? Because internally we don't want to be asked. Do you understand the thing? So basically, if I am not asking question, you will become very vulnerable if somebody asks you a question. So to come out of that vulnerability, I have to prepare myself to by asking question. Do you understand? It's a reverse psychology. Okay, now don't ask me any question because I don't ask any question. Okay, that's what that you want to be in that zone all the time, because I don't want anybody to ask me question. So don't ask me a question. So how do you prepare? You have to ask the question. Then you are making yourself ready to be challenged by others. You get my point? So you have to ask the question. This is the platform for it. So next 4 presentation, we are going to keep quiet. All right?
okay.
All right. Say yes. Okay.
Good evening, all. Actually, my topic is influence of plant spacing on growth, yield, and quality of PKM1 annual Moringa olifera. Now, the general introduction about Moringa. Actually, Moringa is called as a miracle tree. It's indigenous to India. No, the moringa is actually the edible part. It can be used a lot. Leaves, coming to leaves and seeds, and then parts all are edible. In that, moringa leaves are rich in antioxidants, amino acids, vitamins, and so we can say it's a pack house of all nutrients and minerals and rich in antioxidants. So that there is a need to increase the productivity of moringa leaves. So that to increase the productivity, the one of the option is to give the high density planting. So by close spacing, we can increase the productivity, and that was the experiment done here. four different spacings has been taken. First, actually the last three spacings, it's include the double H planting system. There's a 1.50 meter by 0.25 meter, and then 1.50 meter by 0.50 meter, and then in the double H system 1.50 meter by 0.25 meter by 0.25 meter, and then 1.55 meter by 0.5 meter by 0.5 meter. Then this is the field view, and you can see the four different spacings provided in the last two. We can clearly see the double H planting system. In first two spacings we can see only a single row. And then this experiment was conducted in HC&R periculum with three replications, and design used is RBD. Coming to the results, the many parameters has been worked out on growth, yield, and quality. Coming to growth parameters, such as the plant height and number of compound leaves is increased in the closest spacing. Since the plant increased plant height may be due to that etiolation character, since in closest spacing the plants will be there is more competition for light. Here the plant height, so that it can be increased in the closest spacing, while that of number of primary branches and stem girth, the wider spacing has recorded the maximum value, since there will be no competition, and the plants can effectively utilize the nutrients available. And then coming to freshly yield, freshly filled per plant, we can see that the wider spacing has more yield per plant, whereas coming to in hectare, that is the productivity. Here the closest spacing is recorded the more freshly filled and the dryly filled per hectare, since it will accommodate more number of plants in a unit area. Then coming to the quality parameters, first one is vitamin A and ascorbic acid. In vitamin A, we can see that the wider spacing has recorded more maximum value, and ascorbic acid also wider spacing has recorded maximum value. But we can see that manganese and zinc are all highest in the closest spacing, since because there will be more enzymatic activities present here in closest spacing. We can record this zinc and manganese high in close spacing. Also we can see that since plant height is higher in the closest spacing, then it will be related to that auxin, the etiolation character. Zinc is one of the thing which is the precursor of auxin, so that zinc and has recorded maximum in the closest spacing. And coming to iron, iron has recorded maximum in the wider spacing. We can also say that the chlorophyll content is more in the wider spacing, since it receives more light, so that iron is more in the wider spacing. And coming to crude fiber and crude protein, the wider spacing has recorded the more amount of crude fiber, while in crude protein the closest spacing has recorded more amount. Here we can see the freshly filled and the dryly filled per hectare. Okay, here we can see that the closest spacing has recorded more yield. Then this discussions. This we can see that the best treatment, that's the closer spacing. The harvesting has done using leaves are harvested by using a pruning shears. This is a mechanical harvesting. Since we are going for high density planting, the manual harvesting will be more difficult, since there are accommodating more number of plants, so that we are going for this mechanical harvesting, so using some pruning shears. And then a conclusion, we can see that all the micronutrients is highest in the closest spacing.
Any observation related to the leaf extract contents? Because nowadays moringa leaf extract was going to be a point of view for chemical alternative for the chemical treatment.
Yes.
Anything related to that we observed while
no, in this here in this study we have seen only on the yield and some quality parameters. Based on this, it can be continued further to see the how much the leaf extract and processing all we can see further. In this initial study, to only see the actually yield and then quality parameters has been seen in this study. It can be continued further, so that we can see all the processing operations are.
Thank you.
Thank you. Closest spacing, that is 1.50 meter by 0.25 meter by 0.25 meter, sir, so that we can have good yield per hectare, sir. we can also increase the productivity and also micronutrients all we can get high in the clothes. Yes, sir. Double hedge planting, sir. Yes, sir. Sir, between the plants, in actually in a one row, between the plants and then between two rows, it is 0.25, sir. This 1.50 indicates that between two hedges, it is 1.50. Sir, in one row, there will two rows of in one hedge there is two rows of planting. Yes, sir. Two rows, sir. Yes, sir. There is two rows, sir. In that, the two rows distance will be 0.25, and then the distance between the plants will be 0.25. The 1.5. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Ah, between two, sir. Yes, sir. Between. Yes, sir, in a hedge there will be two rows, sir. So that in the two rows distance will be 0.25, sir, and then this 1.5 represent the such as the different treatments distance. Actually, one row, one hedge has been completed. The two rows has been completed, and next hedge for the two rows it will be 1.50 meters. Yes.
Sir, whether any other soil analysis is taken in the study, like how much amount of nutrient that taken by the plant in various spacing?
Uh, Actually, initially a soil sample has been taken and it has analyzed. And it has analyzed for well pH, EC, and then the nutrients. And also after the final results, it has been a soil has been analyzed. But the nutrient uptake studies has not been done here. But the soil sampling has been done and it has been analyzed both before the experiment and also after the experiment.
Thank you, sir.
Thank you. Thank you.
You have done the spacing studies, no?
Yes, sir.
There'll be a competition for nutrients.
Yes.
Have you applied any extra nutrients other than the recommended dose?
Yes, through foliar through fertigation, I have applied the nutrients. Mainly NPK has been supplied. For since it's for a leaf operation, nitrogen has been supplied in large quantities through fertigation.
What is the pruning interval for leaf production?
Leaf production actually first initially from the date of sowing we can go for first harvest in 90 days and then subsequently we can go for 30 to 45 days.
That is very nice presentation. And
Thank you.
I just want to add up, I want to say that, did you analyze the soil microbial activity? That means it will, there is in many research that microbial activity also is very much helpful or increasing the yield. Did you come across such research or some experiment like that?
Oh, only micronutrients has been analyzed in the soil. No microbial study has been done. However, it can be done further, and then microbial activities can also be recorded in further studies.
Okay, thank you, Siddha.
Thank you.
Presenter number 8.
Good evening to one and all present here. I am going to discuss about the topic on performance assessment of Palak in modified nutrient film technique. Introduction. Nowadays, the reason towards cultivation of leafy vegetables has been increased as it having a major role of nutrient. It contains more minerals and vitamins. As per the ICMR recommendation, we have to consume 125 gram of leafy vegetables per day per person. But as the vegetables grown under open field are highly exposed to both biotic and abiotic stresses like unseasonal rainfall, high temperature. So the quality of vegetable production and productivity is decreased as population is also increasing day by day, and also the cultivated land is also decreased by 38.10% due to urbanization. So there is a gap between production and the productivity. So meet out this graph. The more focus should be towards new technologies like a modified NFT, that is nutrient film technique. Nutrient film technique is nothing but it is a hydroponic system. It comes under a closed system of hydroponics. Here the plant's roots are directly exposed to thin film of nutrient solution. Here the nutrient solution is placed. The plants through the permeable roots absorb the nutrient solution. After all the roots have been absorbed, excess nutrients will be again drained out to the storage tank. My aim is to examine the effect of different hydroponic systems and different nutrient combinations on growth and yield of palak. Here I have used FRBD, that is factorial RBD, with 3 replication. Yeah, here I have considered 2 factors, that is factor A as growing system, factor B is different nutrient combination. Here I have used horizontal type and vertical A type. These are the 3 nutrient combinations I have taken. One that is treatment 1 is below the recommended dose. Treatment 2 is recommended dose. Treatment 3 is above the recommended dose. Kg per hectare, sir. Yes. The nutrient quantities were computed for 100 liters of water and was given through fertigation. Throughout the experimental study, the pH and EC was maintained continuously. These are the substrates used in hydroponic studies. This is clay pebbles and coir pith and hydroponic parts. This is a layout of vertical A-type NFT. Here there will be a continuous flow of water through the inlet pipes. After it reaches the roots of all the plants, through the outlet pipe, it again drain back to the nutrient tank. This is a flow of nutrient solution in vertical A type. In horizontal type, the separate tanks for inlet and outlet. Through the inlet tanks, the water will flow and it again drain back to the nutrient tank. By using the system, we can save water up to 90%. It is efficient use of fertilizers can also be done through this NFT system. The effect of different treatments on growth parameters of palak. The plant height, root length, shoot length was maximum under S2 T1, that is vertical A type, along with below the recommended dose of nutrient, because in hydroponics there will be continuous flow of nutrient solution, so the plants can get adequate nutrients. Excess nutrients leads to reduced growth. Low concentration itself enough for plant height and root length and shoot length. Effect of different treatments on yield parameters of palak. The number of leaves is very important as it is directly involved in photosynthesis. As plant height increases, it produces more number of leaves. It directly ultimately result in increased fresh weight. Another parameters are leaf area, leaf chlorophyll and vitamin C. Leaf area, it is directly involved in photosynthetic activity. The leaf area meter was measured using leaf area meter. It was measured at 45th day and the 60th day. Leaf chlorophyll was measured using a spade meter. Vitamin C content, all those was best under vertical A type along with nutrient concentration of 50-50 kg per hectare. Effect of different treatments on quality parameters of palak. Here we have calculated the nutrient content of nitrogen, phosphorus, potassium, calcium, magnesium. This was also best under vertical A type along with treatment 1. This is a growth of palak under different NFT system along with different nutrient solution. The S2 T1 is performed better. Performance of palak under horizontal type and vertical type. The results of the study inferred that the palak grown in vertical A type supplied with NPK at 50-50 kg per hectare showed better growth, yield, physiological and plant growth attributes, nutrient content uptake. I conclude that though the hydroponic is an emerging sector, the hydroponically grown plants can grow faster when compared to soil grown plants. We can produce year-round production. We can meet out the market fluctuality through this.
Very good presentation, Ka. But my question is that when you go for a bigger sized pot, whether any leaf production, the area of leaf production and the total leaf production will be getting increased or not, Ka? Whether any research is going on or further studies have been placed on the size of the pots.
In hydroponics, there are different types are there. There are 6 types, F1 blow system, brick system, drip system, or different types are there. Here I have taken only NFT2 system. We have used only these 2 pot size only.
Very nice presentation, Akka. It is highly advanced. Apart from palak and any other vegetable is that you have tried or it could be possible.
I have tried on mint, palak, and lettuce. I have already presented on mint, so I have chosen for palak.
Whether any diseases observed in this cultivation?
Diseases, I didn't observe any diseases. Pest, whitefly I have observed because previously they have planted cabbage, so I have observed only whitefly, not any diseases.
Akka, whether it has caused any economical damage?
No.
Akka, what kind of protected structure was used in the study, and whether the protected structure will have a significant difference in the yield?
Definitely, it will cause a difference because I have gone for open field polyhouse and also NFT. It is NFT under polyhouse structure. In polyhouse, in lettuce, I have faced the problem of etiolation due to high temperature. Definitely, it will impact on yield.
So I have a question. So which type of water you used, like RO water or normal bore water?
I have used RO water.
Okay, Ka.
While using a normal water, I have faced the problem of high pH and EC. So I have faced many problem while I'm raising the first crop. So many plants have been died due to high concentration of EC. So I have chosen RO water.
In our campus, we have a hydroponics unit. So in that also we'll be using RO water, and also our bore water is highly saline in Trichy condition. So will you recommend any other treatment so that I can treat the water for better utilization of hydroponics?
I will suggest weekly once you have to check pH and EC. Otherwise, before day itself, you have to fill what are the quantities of water you have to use. In my study, I will before day itself I also did, but it is also not good, so I went for RO water.
Thank you.
So for large scale, I will suggest horizontal system with artificial lightning. I will suggest. For large scale means I will suggest horizontal type with artificial lighting. I will suggest. Sir, in a horizontal, the plant population will be less. Sir. In vertical, the plant population will be more, sir. Because in horizontal, we will go for single tier, sir. In vertical, we will go for 3 or we can go for 4, up to 4 tiers. We can go, sir. In case of horizontal, the per plant yield will be more means also. In vertical, 3 tiers are there now, so it will more difficult to compete on yield, sir. So in horizontal type, along with artificial lighting means we can match it with this.
It's a nice presentation. You said whitefly incidence was there. So since we know that this economic part is leaf, we cannot go for inorganic spray.
I have sprayed neem oil.
Okay, only neem oil, or you give some suggestion?
No, I only used neem oil.
Thank you.
Sir, I have clay pebbles, sir. Yes. Yes, sir.
It's a normal recommendation. But in your treatment, you fix it 50, 50, 50, 60 and 70.
Yes, sir.
Why you and reduce the dose actually.
Sir, because in hydroponics there will be continuous flow of nutrients, sir. So less quantity itself enough for the plant growth, sir.
That's why I'm
By
asking.
using this, we can save the nutrients wastage, sir, and the water wastage also we can reduce in this.
That's why if you reduce the quantity,
Yes,
you
sir.
can get better yield, no? So same
Yes, sir.
yield you can achieve.
Yes,
You can
sir.
reduce the fertilizer quantity, no? Then why you fix the same higher dose?
Sir, I have to compare, sir. In excess dose, what is happening? In lower dose, what is happening? To compare, sir.
Okay, but you take only the recommended dose and above the higher dose only, not you are not reducing the dose. Okay.
Sir, actually, my treatment 1 is lower dose, sir. Treatment 2 is recommended dose, sir. Treatment 2 is recommended, sir. 60, 60,
But
60.
you told 50 is recommended.
No,
dose.
sir. Lower dose, sir.
Then it is
50,
okay. So interaction is good. Students are now started asking questions, but better you just stop akka, tangachi, everything. Okay. Not bad, but the same thing is you have to stop.
A very pleasant evening to the dignitaries on the dais and my fellow seniors and classmates. Now myself going to present on the topic post-harvest treatments for improving the shelf life of obtainable papaya coate. In this topic, I have done a research in for a farmer-friendly approach by just investing a single rupee by to increase his shelf life from 3 or 4 days to up to 9 days. This is my result of this research. Now I will go upon the following slides. Introduction about papaya. Papaya is normally called as Carica papaya, belongs to the family Caricaceae and its botanical number is 18. It is rich in vitamin A, that is, it contains about 2020 international units per 100 gram of papaya. It is also a delicious and nutritionally rich fruit, and it is used for the dessert purpose such as candy, tutti frutti, marmalade, nectar, wine, syrup, and for their chromosome numbers. Chromosome number is 18, sorry. Next one, it is a papain. Papain is a protease enzyme which is gaining popularity in the cosmetic and commercial industry such as cosmetics, pharmaceutical, tanning, brewing industry. Next, when we come on to the area and production, the total area under production is 1.44 lakh hectares and the production is 57.80 metric tons and individual productivity is around 43.3 metric tons according to the database of 2019 to 20 from National Horticulture Board. The major papaya growing states in India are Andhra Pradesh, Gujarat, Karnataka, West Bengal, Madhya Pradesh, Maharashtra and Tamil Nadu. The leading countries are India, Brazil, Nigeria, Indonesia, Thailand, Taiwan and Mexico. Now we come into the crop. The crop is a year-round production with high productivity. It will give better economic returns and high nutritional and medicinal value. It produces around, the total crop period is around 22 months and the productivity is around 200 tons per hectare. It is a shallow rooted crop, so it needs adequate nutrient supply for the consistent yield throughout the crop period of 2 years. Now the improved production techniques with the balanced nutrition are also adapted. In papaya cultivation, the major issue is the papaya ring spot virus, which is a devastating virus in the papaya field condition, which is transmitted by aphids, and it leads to lowering the production of 50 to 70% yield loss. Now, this is the treatment details for the post-harvest study, which has been giving as a result. In this, I have given 10 number of treatments, and the design here used is FCRD. And the number of replications is 3 replication, and total number of fruits per replication is around 5 fruits per replication. So totally the different 10 treatments have been discussed as PHT1. P1 means the first factor will be the post-harvest treatment, and second one, the P1 denotes the packaging treatment. I have here used 2 types of packaging. One is without wrapping the fruits with newspaper, and the second type of packaging is with wrapping the fruits with newspaper. That PHT1 is a fruit which is the fruit does not treated with any water, that is the treatments. Then the treatment 2 will be dipping in water, and the treatment 3 will be 2% calcium chloride drip for 2 minutes, and treatment 4 will be 0.1% bevistine for 2 minutes, and treatment 5 will be 2% calcium chloride and 0.1% bevistine for 2 minutes. The total time period for dipping of the fruit is just totally of 2 minutes for individual treatments. Now from this I have came to an inference with the shelf life parameters. That is, first one, we go for the firmness of the fruit. The lowering firmness was observed in the treatment. I have continuously observed for 3, 6th and 9th day. The lower observed treatment was observed in the pH T1 P1, that is the packaging treatment, that is without wrapping and without dipping into any solutions. That showed the lowest firmness of 4.02 in the 3rd day, and in the highest was recorded in the pH T2, pH T3 and P2, which recorded as 4.98. And we go for the 2nd day, which also giving a promising result in pH T3, that is the post-harvest treatment of 2% calcium chloride dip with the packaging, which shows of 2.98. And with the 9th day also, it also confirms the same treatment with the recording of 2.35. So as a result, when we have to give to a farmer, we can say that the firmness has been increased when we go for a treatment of 2% calcium chloride with wrapping of newspaper. The next one is the physiological loss in weight. The physiological loss in weight also says the consistent result as in the treatment pH T3P2, that is the 2% calcium chloride dip plus newspaper wrapping, which shows the lowest physiological loss in weight. So the fruit gets ripened in a very lower period of time. The next one is the days to ripening. The days to ripening shows that the treatment pH T2, pH T4 and P2, that is the packaging treatment, had showed as 9.25, but which is on par with the treatment 3, which is our best treatment, that is 2% calcium chloride with newspaper wrapping. When we come on to the anthracnose incidence, our results will be somewhat negligible to the pH, the treatment of treatment 4, that is the treatment with 2% bevestin with newspaper wrapping showed the higher result. From this is the different photographs of the treatments that is observed on the 3rd day, 6th day and 9th day. From these photographs also we can easily observe that in the 9th day, the first fruit from the left is the fruit that is not wrapped and just kept, and the second fruit is wrapped, and the third photo is the fruit dipped in water and just kept, and the fourth photo is the fruit dipped in water and wrapped with newspaper, and the fifth photo gives the fruits not dipped in water, not dipped in calcium, just dipped in calcium chloride and kept, and the sixth photo dipped with calcium chloride, wrapped with newspaper, and the sixth and seventh photo is dipped with 2% bevestin. First one is not with newspaper wrapping, and the next one is newspaper wrapping. As such, the treatment schedule continues. From this, we can conclude that the treatment pH T2, T3, P2, that is 2% calcium chloride dip with newspaper wrapping, shows the better in increasing the shelf life parameters when we go for the farmer recommendations. From this, the conclusion says that enhanced defense activity because of using of bevestin, and high growth promotion when we go for high nutrient application, and next one is the less disease reduction with the bevestin and newspaper wrapping with calcium chloride. And the fruit, when it comes to ripening, because of the calcium chloride dip, it increases the shell wall thickness, so it gets ripened very slowly.
A nice presentation, sir. Before starting, you said it's a farmer-friendly application. It's a 1 rupee application. In what manner you mentioned that 1 rupee, sir?
Because nowadays the farmers are just wrapping the fruits in newspaper and they are loading into the vehicles. So they're using the newspaper wrapping. Before that, when they are going for just a calcium chloride dip of 2%, it will be more economical. But because the papaya fruits in the market are getting totally devastated within 3 or 4 days, when the farmer go for dipping the fruits alone in the calcium chloride and after that they are wrapping, means the shelf life of the fruit has been increased to 8 or 9 days. So by this way, the farmer can give to the supermarkets or the markets, and the fruits can have a good shelf life, which increases the customers to get the fruits in a high way.
So whether using calcium chloride may cause any health issues?
No, only we are using just 2% calcium chloride dip in 10 liters of water. When we go for any fruit sample analysis, there will be the availability of calcium. The availability of calcium does not raise to a greater extent. Only 0.03% it has been raised when we given for analysis, sir.
Okay, sir. Thank you.
It was a nice presentation, sir. As you said, calcium dip will enhance the shelf life of papaya. So, what about that maximum residual limit, and also in how many days I can consume the fruit after dipping?
After dipping, we can go for consumption immediately because that is not having any health hazard in the calcium chloride dip, because all the fruits will have the calcium content. So only I mentioned that 0.3%, 0.03% only it is increased in the calcium content. So we can consume immediately after they are after the dipping.
This is post-harvest application. Whether any other treatment with pre-harvest application?
I think one research has been conducted in TNAU during 2015 by Monica et al. In that they have gone for the calcium chloride 2% in a foliar spray. That has given an initiation to my work that when we go for a post harvest dip, what will happen? That is the result came here.
Okay, thank you, sir.
Changes in the taste of the fruit.
No, ma'am. I have given for the organoleptic test to all the students. There is no any change in taste, ma'am.
You observed any color changes because you wrapped with newspaper, another treatment is not wrapped?
Yes, when we go for newspaper wrapping, the color of the fruits will be somewhat lighter. When we go for without wrapping, the fruits color will be somewhat darker. This is due to there will no light interaction after covering the fruits, sir.
So because consumer preference that quality of the fruits, that attraction. So only I'm asking.
When we go for that wrapping also, sir, not to a greater extent it given the color change, sir. Only a limited extent, sir. When we go for the fruits without newspaper wrapping, there will be more incidence of anthracnose, sir. That will cause consumer preference. That will not cause the consumers to buy fruits, sir. So in this case, we can go for newspaper wrapping. The color of the fruit will be returned as same as the unwrapped fruit, but just a bit it will be getting reduced, sir.
Which TSS content in which fruit it's more, whether wrapped or not wrapped fruit?
Sir, TSS content also has been enhanced in treatment T3, sir. Since we are wrapping the fruits, no, sir, automatically there will be no light interaction. The fruits get ripening from low days to high days, sir. So automatically when the 9th day, the TSS content of the treatment 3 will be high, sir, than the other three. Foliar spray.
Foliar spray of calcium chloride better. Here dipping, which one is best?
Sir, when we sir, shelf life they also recorded of 7 to 8 days, sir. But when we go for calcium chloride dip, there will be a more amount of nutrients will foliar spray formulations will be in a higher quantity, sir. They will be going for around 200 liters per acre, sir. But when we are taking only 10 liters, sir. So it will be easy for a farmer to do this, sir. Because the operation cost also is high, sir.
But shelf life wise?
Shelf life is this is 9 days, sir. That is 7 days, sir.
7 days only. But here in treatment, you mentioned Bevistin.
Yes, sir.
So we go for Bevistin is a trade name only.
Yes, sir.
Right? You have to put only carbendazim.
Okay, sir.
Good evening, all. Myself Mugilwas. I am pursuing second MSc Forestry under Forest Products and Wildlife Department from FCRI Metupalayam. My study on water quality assessment and its relationship with avian diversity in Navy Limited Corporation (NLC). First of all, in India, according to BNHS, Bombay Natural History Science, they recorded a total of 1,266 birds, in which 57 birds are endemic and 85 species are threatened and 302 species are water birds. There is a saying according to Thiruvalluvar, Nerendri Amayudu Ulage, it means water is the most undoubtedly important parameters which should be used to study the avian diversity. Then why we are studying about avian diversity? Because avian diversity is the basic and important function for conserving a preservation, a biodiversity or an ecosystem. Then my study is mainly focused on the impact of human effect or anthropogenic effect on avian diversity. Then this is my study area, NLC Navy Limited Corporation. It is a central public sector undertaking under the administrative control of Ministry of Coal, Government of India. It was the coal in my Navy first found by T.M. Jabalingam during 1956 in his own field. Then in case of mining, there are two kinds of mining, open cast mining and closed mining. Navy follows open cast mining. There are mainly three areas inside the mining area, that is mine 1, mine 1A and mine 2, in which mine 2 consists of large area. These are the three objectives followed in my study. For this objective, I collected 5 water samples from above mentioned these 3 sites. That is, from mine 1, I collected 2 water samples. That is mine 1 Deer Park Lake and from mine 1 Township Lake. Then from the second section, that is mine 1A, I selected mine 1A Lake 1. I collected water sample from mine 1A Lake 1 and mine 1A Lake 2. Then from the last mining site, I collected the water sample from mine 2 Viewpoint Lake. And for comparison to the water quality with these water lakes, I collected the water samples which is present in the Neyveli township, which is the original water sample which is present in the Cuddalore district. So by collecting the water samples, I followed these objectives. First of all, I determined the physiochemical properties of the water sample, then elemental analysis of the different water parameters. Then I correlated these water quality parameters with avian diversity. So for surveying birds, I followed point count method. By the picture, we can identify that the birds are collected and noted on the records by standing in a fixed position, and the birds which is surrounded by the observer are collected and recorded. Then I done my survey during March, May, that is summer. Then these are the different water quality parameters which I selected for determination, that is pH, electric conductivity, then dissolved oxygen, BOD, elemental analysis, then chlorides. And these are the different methods which are used for determining these parameters. For pH, I used potentiometer methods. Then for determining electric conductivity, I used conductometric methods. Then Winkler method for dissolved oxygen. Then for BOD, I used BOD determination without dilution method. And for elemental analysis, I used atomic absorption spectrophotometer. Then for chlorides, I used Morse method. Then the results found that I recorded totally 30 bird species inside the mining area, in which I recorded total of 983 count. By comparing the different water sample which I selected from inside the mining area, I got to know that the bird diversity is very rich in mine 1 Township Lake, and it is directly correlated with the it is only directly correlated with the pH and DO which is present in the water. The other elemental factor or physico-chemical factors are indirectly correlated with the water sample. So I saw maximum avian diversity in mine 1 Township Lake and minimum in mine 1A Lake 2. Then this is the correlation table which I recorded. In this, we can clearly see that pH and DO only directly correlated with the bird diversity. So the conclusion of my study is water quality is an important factor which determines the abundance of wetland birds. Then by enhancing this water quality, we can promote the biodiversity, avian biodiversity. The major methods which we can increase the avian diversity is by the major method we can apply for improving the water quality is by the afforestation, because inside mining area there will be less afforested area, so the mining diversity will be also reduced. So by increasing afforestation areas inside the mining area, we can increase the avian diversity inside the mining area, and we can recover that area to a good ecosystem.
Is there, have you recorded any migratory birds visiting this mined areas? Because it's already deforested or already scraped land. So, what's your take on
Totally, I recorded 30 species, in which 3 species are comes under migrant, which is darter bird or oriental white ibis and painted stork. These are 3 migrant birds which I found in mining area during summer.
This was a nice presentation, sir. Which species is coming? Number of population will be the more in that particular mining area.
Totally, I recorded 98 species, in which maximum number like about 250 was counted for spot-billed duck. It is very common wetland species which is seen in Cuddalore district. Yeah, the hardness of shell is very less. Yeah. because of the calcium content is very less. Yeah.
It was a nice presentation. Other than March, May, in any other seasons, how about the birds population and survey?
Actually, I actually studied 3 seasons comparatively: summer, monsoon, and winter. In comparing this season, I collected more number of birds in this season only, summer. That is why I presented here.
Why I asked the doubt, as in summer, the water content will reduce.
Yeah.
As for other seasons, the water content will be more. And the residual effect or any other, it will reduce due to the high water content. So I asked how about the birds.
But
So it was
but
in other seasons.
But inside the mining area, it is different. Like migratory birds are coming randomly. Like migratory birds are coming more during the summer season. That's the main reason. Because I told like 3 migratory birds are coming. Other than the other season, only 25, 24 kinds of species are seeing in other 2 kinds of season.
Thank you.
Other bird population, do you study any other related to fish other than insect related to water? Why? Because that birds are coming to eat that fish only, fish insect. Do you count any population related to fish?
Not counted, but I saw fishes inside the lake.
Fishes are there.
Yeah, I saw that, but I not took any survey on that.
Microbes, other than microbes, insects.
No,
Because
no.
birds are.
Yeah, I understood. But I don't. But I didn't do any survey on that thing. That's
It's a I don't know what happened after the break. We just put something in your mind, or it's the impact of the tea on the snack. It becomes very lively, and this is how you have to go, and this is the whole purpose of it. It's not like just presenting and listening to that. The question you ask, the deeper you get an understanding, and it also helps the speakers. You know, they spend a lot of time in collecting data and so now when you ask the question, if you ask 3 or 5 3 to 5 questions, if they answer 80% of that, they feel immensely satisfied and their confidence will be get a boost. So you are actually indirectly helping them, right? Okay. Yeah, actually, the philosophy of science talks about correlation and causation. You studied your water quality parameter, then you put your correlation. You are not saying that water quality is the cause of the bird diversity in that area. Is that correct?
Sir.
So you put a correlation between
Yes,
water
sir. Yes,
quality
sir.
and the biodiversity. Are you trying to say the water quality is the cause of the richness of the diversity? No, right?
No, sir.
All right. Okay. So you have to be very careful when you run a correlation. You try to match up what's the relationship between the parameters, but it is not the cause of it. It's not a causation. So it's a very good example that it's very difficult to connect the water quality deterioration from your study and connecting to that the diversity of the bird species. So a lot of things has to be done, and in wildlife research, it's very difficult. The pollution stress studies are very limited, and because the wildlife are moving from place to place, as somebody was asking, the migratory species are involved. So definitely the exposure to a particular site which is contaminated like yours is very rare because these are mobile, they are not plants. You understand? So that's a very important thing. And it's very interesting because yours is completely out of the equation of all the previous presentation. One more thing before we close, I think the good thing about asking the question, if you have a problem asking the question, then make a statement before you ask the question. First, appreciate the presentation, just speak 2 or 3 statements, then question automatically will come. It's always difficult to ask a question, right? So only few of you have raised, I mean, started like the presentation is good, even it is a lie, just do it. Yeah, because you have to know how to ask question before you. If you don't make a statement, then question becomes very difficult. So just 2 sentences is about your slide is very good. Just talk, then the question will become easier. Okay, so that's a good practice that you have to make. All right, so we'll stop here.
To the panel for the concluding remarks.
So, chairman and then co-chairman, then rapporteur, fellow students, as well as campaigners. So it was a nice, session. So we had a very good presentation by our chairman, Dr. Hamid Sulaiman, who has presented a very good presentation of the environments, and it was followed by 10 presentations in diversified topics. I appreciate the people who have selected these oral presentations. So we had a diversified topics in forestry, in banana, in papaya, in moringa, and even in palmyra, and also in floriculture, that is canna indica, and also in different crops. So we had a very diversified presentation and also diversified works, starting from hydroponics to nutrient status, as well as other breeding work and other things. So this is a very good start for, I can say, I congratulate the students. So you can have this type of programs, because we have been involved in this, organizing this agricultural graduate conference. Maybe I am involved for the past 8 years. So this is the 8th conference we are attending. So the conference pattern as well as the style of functioning has improved a lot. Now you know that the skill has developed, and you know how to do a presentation, and also you have to make within the time limit, and what you want to impress upon the audience also you have done very nicely. So I appreciate all the students who have done. I encourage all the other students also who have done the sent the articles, and they have been selected for maybe for the poster presentation. Because of the time constraint, they may not be able to accommodate. And then my piece of thing is, you try to improve your article repeatedly, write, and then try to write it, and then focus on the NAS rated journals more than 6, or international journals, so that you will be able to publish your work. As Madam Vice Chancellor told, we do a lot of work, but we don't publish. So there is a statement that you publish or perish, something like that, and that is more important nowadays. The science lives in the publishing your articles, and then how you are peer reviewed. So our times, no, didn't get this score and all, but anyhow, so we have to go along with the stream. So in that context, I congratulate all the team, all the workers, all the students, and Dean PGS, as well as chairman, and the rapporteur, and all the people for giving us the opportunity. Thank you.
Kindly request Dr. Paramaguru sir, Dean, Horticulture College and Research Institute for Women, to honor our lead speaker with a memento. I request Dr. Muthuvel sir to honor our co-chairman, Dr. Paramaguru sir, with a memento. I request Dr. Chitra ma'am to present a memento to Dr. Muthuvel sir. I request Mr. Gokul, student rapporteur, to present a memento to Dr. Chitra ma'am. Thank you, sirs. Thank you, ma'am. I invite the staff rapporteur of the event, Dr. R. Chitra ma'am, Associate Professor, to deliver the vote of thanks.
Good evening to all. So this is the right time to propose a vote of thanks to our delegates as well as the students. So first, my heartfelt thanks to our Professor and Head Department of Fruit Science, Dr. I. Muthuvel sir. Sir, thank you very much for your introduction speech to our speaker. Next, I thanks to our Dean, HCNDR Women's College, Trichy, Dr. P. Paramaguru sir. So, in this in his speech, he motivate these students how to write the articles and how to publish the articles more than 6 NAS rating. So that is useful for your future also. So thank you very much, sir. So it is a this is not our thanks. This is our gratitude to on behalf of Vice Chancellor and Dean SPGS, Dean Horticulture, to our lead speaker, Dr. Hamid Sulaiman, Associate Professor, Department of Environmental Science, Oman. So, sir topic is very different. So we are the students from horticulture only. So we are knowing about the fruits, vegetables, spices, and flowers, medicinal crops itself. So sir's presentation is very different. So that is the base material for our future research also. So thank you very much, sir, for your kind and brief presentations, which is helpful for our future research work. It is not for students, and our staff also propose a project related in this aspect, sir. Thank you very much, sir. So here, followed by the lead speaker. So 10 presentations are presented by the students from different faculties. So I thank you very much, each and every students, because students is stick on the time within before that program. They mentioned that only had at least only 5 minutes. So the students stick on the times is only within 5 minutes. So thank you very much, each and every presenter. And finally, I thank to the all the students is assembled and occupied in the areas. But now the most of the areas are vacant itself. So it is mainly due to the time itself. So once again, I thanks to all the students, organizer, Dean SPGS for conducting this great conference, and also permissions is already given by our Vice Chancellor. So I thank to our beloved Vice Chancellor and our Dean Horticulture and Dean Horticulture Periukulam and Dean Horticulture Trichy. So finally, I thank each and every one of the people involved in these occasions. Thank you very much. Thank you.
Thank you, ma'am. I kindly request all the presenters to gather in the dais for a group photo with the evaluation committees are available on the table outside. Please collect it while you go.